Welcome to the 2026 Annual Convention of the PCHRD Scholar Society. I'm Brian Andrich L. Paulo, MD PhD candidate and your oral presenter. The title of my study is Diagnostic Accuracy of an Immunoassay Using Avidity-Enhanced Polymeric Peptides for SARS-CoV-2 Antibody Detection. This video details the development of a synthetic peptide-based immunoassay for SARS-CoV-2 antibody detection. Our team includes collaborators from the Biomedical Innovations Research for Translational Health Science Laboratory, the Institute of Clinical Epidemiology, DOST PCHRD, and the Department of Medical Microbiology at the College of Public Health. This is part of my dissertation defended last February. This study addresses a major challenge in synthetic peptide diagnostics, which is how to reproduce the strong antigen-antibody binding of full proteins using shorter and simpler molecules. Synthetic peptides are being developed as alternatives to protein antigens because they allow precise targeting of epitopes, improved stability outside cold chain conditions, and simpler manufacturing without biological expression systems. However, peptides usually have fewer epitopes than whole proteins. This leads to weaker total binding strength, or avidity. Improving avidity is therefore the key limitation that must be addressed. This study aimed to enhance peptide avidity through polymerization. I prepared immune complexes between polymeric peptides and patient antibodies and evaluated their diagnostic performance. The long-term goal is to develop cost-effective and scalable alternatives to protein antigens. We retrieved SARS-CoV-2 epitopes from the immune epitope database and grouped them into clusters. We designed cysteine-flanked peptides to allow disulfide-mediated polymerization. We compared polymeric and depolymerized forms to determine whether avidity improved. Binding was measured using enzyme-linked immunosorbent assay and affinity estimates. The top peptide was developed into a diagnostic assay and evaluated for analytical and clinical performance. We designed 15 unique sequences from 15 epitope clusters across 7 structural and non-structural SARS-CoV-2 proteins, supported by empirical searching and computational modeling. Quality control confirmed high purity and correct molecular mass. These are the three-dimensional space-filling models of the proteins. The model on the white background is the spike protein, and the S559 epitope is mapped in red. We confirmed that our CORN depolymerization method, utilizing N-acetylcysteine, effectively breaks down polymeric peptides into monomeric forms, as demonstrated by the SDS phase results here on the left, and the corresponding densitometric analysis on the right. Lanes 2 and 8 shows the streak or smear obtained with the polymeric peptide, while lane 7 shows that N-acetylcysteine was effective in depolymerization. We wanted to determine whether the polymeric form of the peptide binds more strongly to antibodies. And so for that, I prepared the peptide in various degrees of depolymerization. In ELISA results here on the left, the completely polymeric peptide is shown in red, labeled 0% acetylcysteine. These immunoassay results were then taken and linearized using the Liliom method to come up with estimates of the binding affinity. For instance, in subfigure B, we were able to come up with an estimate of the polymeric binding affinity at 29.26 nanomoles. However, when the peptide is depolymerized, the dissociation constant increases to 63.76 nanomoles. And because the relationship is inverse, higher dissociation constant means lower strength of binding, then that means that depolymerization lowers binding strength. Therefore, from there, we could say that polymerization leads to a 2.1 8-fold gain in avidity. Next, we wanted to perform screening of our peptides, and the results are shown here. We tested it on a pooled sample of 20 COVID-19 patients relative to 20 pre-COVID-19 biobank controls. Labeled in the x-axis are the different peptides, and we can see here that the most reactive peptides come from the spike protein. It's also notable that the M212 peptide, despite its intraviral location, was reactive to patient antibodies as well. S559 was the top polymeric peptide in terms of antibody binding, so we developed it further into a diagnostic immunoassay. We wanted to determine the analytic performance of the S559-based ELISA. To do this, we tested it in this setup. We had various dilutions of pooled sera. The red line shows assay signals with COVID-19 sera, while the orange line shows pre-COVID-19 sera. This is a total of 6 technical replicates. We were able to calculate the limit of detection at a dilution of 1:6,400, and the limit of quantitation at 1:1,600. The intra-assay precision, based on the percent coefficient of variation, is between 1.87% and 4.4%, which is lower than 15% across concentrations tested. The signal-to-noise ratio was around 6. Although lower than 10 for experimental purposes, this is acceptable. Taking these results together, one could say that although the signal is weak, it is repeatable. Also from this, we calculated the threshold of the assay at 0.057 absorbance units based on the formula, mean plus 3 standard deviations of the blank readings. Applying the S559 ELISA to clinical Syrah demonstrated strong diagnostic performance. We applied it on 1,222 COVID-19 clinical samples and 218 biobanked controls. In this figure on the right, each dot corresponds to the immunoassay results of a specific clinical sample. These are arranged per severity, asymptomatic to critical. Applying the predefined threshold, we were able to calculate a performance of 83% sensitivity and 97% specificity. If we move this cutoff or threshold further to the ideal post-hoc threshold, using Uden-J, we could come up with a sensitivity of 95% and a specificity of 100%. Receiver operating characteristics, or ROC analysis, revealed an AU-ROC of 96.59% for the polymeric S559, significantly outperforming the M212 and N10 comparator antigens. M212 has a performance of 74.12%, while N10 hectares a performance of 58%. In order to understand these results better, I'm going to proceed with a short discussion. Our S559 assay may have a diagnostic utility. It doesn't need a separate scaffold. Also, our CORN-Liliom approach to estimate binding affinities, an improvement over traditional methods of measuring avidity gain. Traditional methods of measuring avidity involve chaotrope ELISA. In chaotrope ELISA, you add a chaotrope such as urea to try to elute the antibody from the antigen. However, what usually happens is that the antigen itself gets eluted from the bottom of the well, thus leading to false positive claims about avidity gain. Also, for our peptide screening, we found that most of the peptides that are reactive are found on the surface of spike. This is expected because of their surface location and their role in viral entry. But for M212, it is internally located, and we think that this may be a form of decoy, in which the virus tries to mislead or misinform the human immune system. But there is a caveat. Because assay results do not necessarily directly correspond to immunodominance. In order to test positive for our screening, a peptide has to do at least two things. First, it has to bind efficiently to the bottom of an ELISA plate, and the second is that it has to bind antibodies. Thus, for peptides with a negative result, it's possible that the peptide was just not efficiently coated at the bottom of the well. Coating efficiency would depend on various factors, such as hydrophobicity, charge, and folding propensity. In terms of analytic performance, the assay is sensitive and highly specific. Nevertheless, if it falls short of regulatory requirements, there are remedies to that. We could multiplex, combine this test in parallel with other peptide tests to improve sensitivity. Also, it is significant that the peptide has a relatively high sensitivity compared to other peptides, as peptides tend to have lower sensitivity than whole proteins. We hypothesize that this gain sensitivity may be coming from the ability, which in turn comes from polymerization. In conclusion, peptide analogs of SARS-CoV-2 proteins demonstrated antibody binding. Synthetic peptides can recapitulate key properties of whole antigens, supporting their potential for further development into novel diagnostics for COVID-19. Future work should focus on further validation and scale-up. Approach may be applied to other human diseases, particularly in outbreak settings. For a more in-depth look at this research, please scan the QR code to access the preprint. A partial list of references is provided on the left. I would like to acknowledge my mentors, collaborators, and institutional partners. Thank you for your attention.